Oxygen levels play a critical role in the generation and efficacy of adoptive cell therapies, ACT, a form of immunotherapy that involves engineering and infusing immune cells, such as T cells, into patients to combat diseases like cancer and autoimmunity. The significance of oxygen levels in this process can be understood through its impact on cell metabolism, differentiation, survival, and function, all of which are essential for the success of ACT. Oxygen is a key regulator of cellular metabolism, particularly in immune cells like T cells, which are central to adoptive cell therapies. T cells rely on oxidative phosphorylation in the mitochondria for energy production in oxygen-rich environments. However, under low oxygen, hypoxic, conditions, such as those found in tumor microenvironments, T cells switch to glycolysis, a less efficient but faster energy-producing pathway. This metabolic shift can influence T cell differentiation and function. For instance, hypoxic conditions often promote the generation of memory T cells, which are long-lived and highly effective in mounting sustained immune responses. Conversely, high oxygen levels may favor the differentiation of effector T cells, which are more short-lived but potent in immediate tumor killing. Understanding and manipulating oxygen levels during the ex vivo expansion of T cells can thus optimize their metabolic state for therapeutic efficacy. Oxygen levels significantly influence T cell differentiation, which is crucial for adoptive cell therapies. Hypoxia-inducible factors, transcription factors activated under low oxygen conditions, play a pivotal role in shaping T cell phenotypes, acting as master regulators of hundreds of genes. HIF-1 alpha, for example, promotes the differentiation of T cells into Th17 cells, which are associated with inflammation, while suppressing regulatory T cells, also known as Tregs, which can inhibit anti-tumor immunity. For cell therapies, the balance between cytotoxic effector T cells and Tregs is critical. Hypoxic conditions during T cell expansion may enhance the generation of tumor-specific cytotoxic T cells while minimizing Treg activity, thereby improving therapeutic outcomes. The survival and persistence of adoptively transferred T cells in vivo are key determinants of adoptive cell therapy success. Oxygen levels during the ex vivo culture phase can influence the expression of survival factors and resistance to apoptosis. For example, hypoxic conditions have been shown to upregulate anti-apoptotic proteins like BCL2, enhancing T cell survival post-infusion. Additionally, hypoxia can promote the generation of stem-like memory T cells, which have superior persistence and self-renewal capabilities compared to effector T cells. These long-lived T cells are highly desirable for ACT, as they can provide durable anti-tumor responses. The tumor microenvironment, TME, is often characterized by severe hypoxia, which poses a challenge for the efficacy of ACT. Hypoxic TMEs can impair T cell function by inducing exhaustion, a state of reduced cytotoxicity and cytokine production. Exhausted T cells express inhibitory receptors like PD-1 and CTLA-4, which limit their anti-tumor activity. Preconditioning T cells under hypoxic conditions during ex vivo expansion may enhance their adaptability and function in the hypoxic TME. Additionally, strategies to modulate oxygen levels in the TME, such as combining ACT with anti-angiogenic therapies or oxygen delivery systems, could improve T cell infiltration and function. Oxygen levels also impact the scalability and reproducibility of ACT manufacturing. Traditional T cell expansion protocols often use ambient oxygen levels, 21%, which may not reflect physiological conditions. However, physiological oxygen levels in tissues range from 1% to 11%, with even lower levels in the TME. Culturing T cells under physiologically relevant oxygen conditions can improve their functionality and reduce oxidative stress, which can damage cells during expansion. Moreover, hypoxia can enhance the yield of certain T cell subsets, such as stem-like memory T cells, which are desirable for ACT. Optimizing oxygen levels during manufacturing is thus essential for producing high-quality, clinically effective T cell products. The manipulation of oxygen levels in ACT has significant clinical implications. For example, hypoxia-conditioned T cells have shown enhanced anti-tumor activity in preclinical models, suggesting that oxygen modulation could improve ACT outcomes in patients. Additionally, combining ACT with therapies that target hypoxia, such as HIF inhibitors or oxygen-carrying nanoparticles, could further enhance efficacy. Future research should focus on defining optimal oxygen conditions for different T-cell subsets and developing standardized protocols for oxygen modulation in ACT manufacturing. Oxygen levels are a critical factor in the generation and success of adoptive cell therapies. By influencing T-cell metabolism, differentiation, survival, and function, oxygen modulation can enhance the efficacy of ACT and improve patient outcomes. As the field of immunotherapy advances, understanding and harnessing the role of oxygen in T-cell biology will be essential for developing next-generation adoptive cell therapy strategies. Discover ambient solutions for low-oxygen immune cell culture.